Record button. There we go. We've started recording, so if you want to kick off, Greg, then off you go.
Yeah, absolutely. So this lecture, well, first of all, I'm Greg Halliwell. I'm a third-year medic, mainly looking at neuroscience and related things. I'm going to be giving this lecture on neuromuscular transmission and then on to cardiac, smooth and skeletal muscle, the three types of muscle in the human body. It's quite a vast topic in the course. You'll see it takes up a few lectures, so I'm just sort of skimming the surface, the core things today. If you're going to go into detail and write essays and things on this, you might want to delve a little deeper, and I'll give some pointers as to areas that might be good to include in an essay or something like that. So I'm going to start thinking about the neuromuscular junction, and I want us to think about how we need to tell this as a story. It's how an electrical signal, an impulse from the brain down a motor neuron, how that's able to be converted through these stages into kinetic energy, which is the ultimate goal of a muscle. And so in thinking about the neuromuscular junction, we're going to be thinking about this step from the electrical signal to the chemical signal to the electrical signal. And then when we get onto the muscle, we'll be looking at how the electrical signal that's created at the neuromuscular junction is converted into kinetic energy. So some key terms to start with the neuromuscular junction. If we look on the right, we can see a micrograph I found of the neuromuscular junction with these motor end plates, which are the terminus of the motor neuron branches, onto a singular muscle fiber. And we can see these boutons, which are the bulb-like endings, and they're the points of interface with the muscle. And they sprout into imaginations into the muscle. And all the muscles innervated by a single motor neuron is called a motor unit. And that's important because if we think of what a single impulse down motor neuron is going to do, it's going to recruit the muscles within a single motor unit that are going to work together to produce the contraction or the action. So to tell the story of the neuromuscular junction, there's this diagram from this really helpful handbook, which I'm sure a lot of you will hopefully be familiar with. If not, definitely take a copy of it out. And our challenge is to follow the journey of a molecule of ACH from the moment it's incorporated and synthesized in the presynaptic membrane, and how it's transmitted and how it's received at the postsynaptic membrane. And that's what we're going to be thinking about these next few slides. So the first thing to think about is its synthesis, and it's synthesized from choline, which is recycled, and we'll come to that in a minute, and an acetyl group, which is donated to the choline molecule by acetyl coenzyme A, which is converted back to coenzyme A. And this reaction is catalyzed by the choline acetyl transferase enzyme, which is spelt weirdly there. I don't know why. But then this is the molecule of acetylcholine with its positive nitrogen group, which is important. And then, so once we've made our molecule of ACH, we need to load it into a vesicle. And that's going to enable us to have the quantum release that we'll come to in a minute. And this uptake into vesicles is done by this the vesicle acetylcholine transporter. And this utilizes a gradient that's been set up by other molecules, set up by, for example, the proton pump, the H plus ATPase, and the exchanges acetylcholine for protons, and utilizes that gradient to load it into the vesicles. And the significance of the vesicles comes into play when we have our signal from the motor neuron. So we have this store of vesicles containing ACH that are ready and releasable and ready to go. And we get an action potential coming into the presynaptic neuron, the signal from the brain that's ultimately going to result in the kinetic motion of the muscle. And we need to think about how all the steps in between, how that gets converted into the force. So this action potential in the presynaptic neuron, the signal, if you like, is converted into this calcium influx This calcium is the messenger, and the opening of these voltage-gated calcium channels allows calcium, but it's important to note that action potentials have to be very discrete. We don't want continued contraction. We want these messages down the axon to be transient and not continuous. So it's important to consider how they're extruded at the end of the action potential, as is by the various transporters and pumps that get rid of calcium from any cell. Once again, it enables the exocytosis of these vesicles into the postsynaptic through the postsynaptic membrane and into the synaptic cleft. And this is actually quite a small number of vesicles that you get released in response to an action potential, only about 200, each containing about 10,000 molecules of acetylcholine. So this is the amount of acetylcholine released from one vesicle is what we consider something called a quantum, and so that gives the term quantal release, which is how we describe the release at the neuromuscular junction. So exocytosis requires vesicles to bind to these snare proteins, and I'm not going to go into great detail on how the calcium influx changes the structure of these snare proteins, but that might be something good to consider if you're considering writing an essay on this topic, because there's a lot of interesting research into thinking about how these enable the calcium sensitivity of the vesicle release. So the vesicles bind to these snare proteins, and then when calcium binds to these snare proteins, they allow exocytosis. And there's a sort of misconception, because vesicles are made of the phospholipid membrane, I think that with every exocytosis, a vesicle is just incorporated into the membrane and new vesicles are made. But if you can imagine, that would be a very energy expensive process. And so we have instead this recycling of the vesicles in this so-called kiss and run hypothesis, where the vesicles kiss the outside, release their acetylcholine, and then the vesicles are recycled. So the acetylcholine can diffuse across the synaptic cleft, and it binds to postsynaptic nicotinic acetylcholine receptors, and they're non-specific cation channels, and they're the key functional molecule at the postsynapse of the neuromuscular junction. And there's a wide range of literature, and they've been very intensely studied because they're very important molecules in the body. And I would encourage you to look at that again if you're thinking of writing an essay or something on this topic. But the influx of these positive ions leads to these depolarizations, these end plate potentials that can form. And if they're sufficiently large, if the amount of quantal acetylcholine release is enough, and the end plate potential reaches a threshold of around -50 millivolts, you get further opening of voltage-gated sodium channels, and that leads to this postsynaptic action potential, which is going to be transduced in the muscle in various ways that we're going to come to. So we don't want this to last forever, and in conditions where it does, we get tetany and complete failure of various body systems. We can't breathe if our muscles are sustained contraction. So we have an enzyme called acetylcholine esterase, and that essentially does the opposite of the choline acetyl transferase we saw before. It takes acetylcholine, converts it to choline and acetic acid. And this is an important molecule for pharmacological manipulations that we'll come to in a minute. And to a lot of this is about conserving, making this an energy economic process. So we recycle the choline, we take it back into the presynaptic knob rather than synthesizing it again. And this is by the sodium choline cotransporter. Okay, so thinking about an example of how we understand the importance of the neuromuscular membrane. And Myasthenia gravis is an example of a disease that's like beautifully shows the significance of a process with a very definitive cause, a very definitive mechanism, and a very intuitive treatment. So it's a really good one to so the core symptoms of myasthenia gravis are weaknesses of muscles in the eye, face, and swallowing, and these are all areas where there's a high sort of drive onto the muscle, and so it presents with drooping eyelids, double vision, muscle weakness, which can be throughout the body, but is particularly in the face and hands. And so its cause is antibodies against the nicotinic acetylcholine receptors. So it's an autoimmune disease, and it selectively knocks out these nicotinic acetylcholine receptors. And essentially, what we get is a deficit of this neuromuscular junction transmission. The presynaptic action potential isn't able to induce depolarization postsynaptically, and so we have these deficits. And the treatment to overcome the fact that we haven't got enough neuromuscular transmission, we need to enhance neuromuscular transmission, and we do this by inhibiting the acetylcholine esterase enzyme. And we do this by neostigmine and physostigmine, and these are transient inhibitors. We don't want to completely block it with high potency, because it's still necessary to stop muscle tetany and muscle. So these are quite low affinity binding. And there are other ways we can use drugs to have desired effects at the neuromuscular junction, and sometimes undesired effects as well. So a classic example is botulinum toxin, or Botox, which you might know for its use in cosmetic surgery, but it's actually a potent toxin, and a potent natural toxin, and it exerts its effect by inhibiting the excitosis of acetylcholine vesicles. And that means it actually can be used to treat muscle spasms, because it stops neuromuscular transmission, and in cases where that transmission is excessive, as in muscle spasms, it can be beneficial to reduce the levels of excitosis. But at toxic doses, then it's going to stop breathing. It's going to stop it's going to cause tetany, and sorry, no, it's not. It's going to stop breathing, but it's going to stop muscular transmission where it's necessary. And also, acetylcholine is important in the parasympathetic nervous system, so we see parasympathetic effects as well. And there's a couple of other experimental drugs that target the presynapse as well, and these are experimental. They don't have clinical use at the moment, but if you're writing an essay on this topic, it's a good thing to bring in these different manipulations of the presynapse and how they elucidate what the function of these various presynaptic molecules and processes are. So in the synaptic cleft, we've already considered neostigmine and physostigmine, which are inhibitors of the acetylcholine esterase enzyme, and so they increase the availability of acetylcholine and facilitate neuromuscular transmission when it's deficient, as in myasthenia gravis. But nerve agents such as sarin and Novichok, which was in the news when I learned about this, they irreversibly inhibit the acetylcholine esterase, and so essentially we get uncontrolled muscle tetany. We can't stop the neuromuscular transmission once the action potential is passed, and so we get widespread effects throughout the body. So targeting the postsynapse is an important target for thinking about anesthesia, because in anesthesia we need to completely relax the muscles. We don't want muscles to be contracting and getting in the way of any surgical interventions or breathing apparatus and things. So as part of that, there's two key types of molecules that interact with the postsynapse to try and relax the muscles, and interestingly they both have antagonistic effects. So we've got non-polar depolarizing blockers and depolarizing blockers, So to start with, the sort of simpler concept of the non-depolarizing blockers, and this is exemplified by tubicurarin, which is from a toxin that used to be used in combat arrows, but has since had therapeutic use. It's a competitive antagonist of the acetylcholine receptors, and it blocks neuromuscular transmission and relaxes the muscles by stopping the transduction of the acetylcholine in the synaptic cleft, stopping that being turned into a postsynaptic depolarization. Tubicurarin is the classic molecule, but nowadays we have the faster acting and less general molecules such as vercuronium. We get these side effects, but we can reverse it using an acetylcholine esterase inhibitor, which makes sense when you think about if we're reducing the amount of transmission we can have with the nicotinic acetylcholine receptors. If we raise acetylcholine in the synaptic cleft, we can start to overcome that. So depolarizing blockers, they do the opposite. They are actually agonists at nicotinic acetylcholine receptors, and so they act to depolarize the postsynaptic membrane. But the key point is they're not broken down by the acetylcholine esterase, so their action is sustained. And this initially causes muscle spasm and tetany, but quickly gives way to a block of transmission because the channels can't recover from an inactivated state. So they have less effect on autonomic ganglia, but the sustained opening of the channel, this leads to changes in potassium homeostasis, which can cause issues. So that's I've just whizzed through the neuromuscular junction, but I think a key point is it's a classic example of chemical synapse. In the brain, we have loads of synapses, but things can be very complex. We get co-transmission, we get sideways transmission, we get all sorts of things. But a neuromuscular junction is a perfect place to study the synapse because it's very simple. We have one neurotransmitter and one key postsynaptic target. It's a clinically important site, and you can modulate it with many drugs. Okay, so I've talked through neuromuscular transmission, and now I'm going to think about, once we've got past the neuromuscular junction, how that's converted to movement via these three types of muscle. And there's a key question I want you to bear in mind about these things, and an important question to address if you're considering writing an essay on this topic. Why do we need three types of muscle? What is the key functional differences that mean we need structural differences in the muscle? Why don't we just have one type of muscle that performs all three roles? Because ultimately they all contract. So first thing, we're going to think about skeletal muscle structure, and we can't have an understanding of the molecular processes that go on without having an understanding of the gross processes and the gross structure of the muscle. So here we can see a muscle, and each muscle contains, is split into muscle fascicles, which are further split into muscle fibers, and this is the cellular level. So if anyone's talking about a muscle cell, they're talking about a muscle fiber. And then, but we can go deeper than that, and we get fibrils, and within the myofibrils are the myofilaments that are key contractile machinery of the muscle. So here we can see a diagram of a muscle fiber, and then inside that a myofibril, and then inside the myofibril we can see the thick myofilaments, which are made of myosin, and the thin actin filaments. And we can see these different terminologies we give to the areas of the myofibril, and the key one I want to focus on is the sarcomere, and we're going to be thinking about how the lengthening and the contraction of the sarcomere leads to movement. So the muscle fibers have the myofibrils and then the myofilaments in them, and the sarcomeres are defined by the Z lines that split transverse across the cylindrical myofibrils. So the myofibrils are formed from these interdigitating myofilaments, the thick filaments and the thin filaments. And so first thing about the thick filaments, we see they have these two intertwined myosin chains, myosin heavy chains with globular heads, and heads bind to myosin light chains, and these are regulatory chains, so they regulate its binding. And connected to the Z line by this key protein titin. Titin is the biggest protein in the human body. It's a massive protein, and it actually, despite the fact that it's very big, it proves very elusive. But now we understand it's got a very key function in how this myosin molecule operates, and it's essentially a spring. It holds it to the Z line, but there's the thin filaments, which are much narrower, and they consist of two intertwined alpha helix actin chains, which we can see in the diagram here. So the key molecules that act with this chain are the troponin molecules and the tropomyosin molecules. And the key role of the tropomyosin molecules, it obstructs the myosin binding site, which is located inside the groove between the two strands of actin. And troponin is a trimer. It's got three constituent parts: the troponin T that binds to tropomyosin, troponin I that binds to the actin binding site, and troponin C that binds to calcium ions. So it enables this calcium sensitivity of muscle contraction that we'll come to in a minute. So how do we get from the depolarization in the postsynaptic membrane to the contraction using this machinery that I've just outlined? Well, we get depolarization of the sarcolemma, which is another word for the muscular cell membrane. And these action potentials spread along the T tubules, which are these invaginations of the sarcolemmal membrane. And these are located at the interface of the I band and the A band. And they interface with sarcoplasmic reticulum inside the cells. And these sarcoplasmic reticulum are the key calcium stores in muscle cells. And they form structures called triads. As you can see, the T tubule comes very close to the sarcoplasmic reticulum, which is intracellularly. And this is important in skeletal muscle when we're considering excitation-contraction coupling, which is how that excitation that travels through these T tubules is converted into contraction, which is the ultimate goal of a muscle cell. So the T tubule membrane has these L-type calcium channels, and they're also called dihydroxypyridine receptors or DHP receptors. And they undergo a conformational change, and that makes them calcium permeable. But in skeletal muscle, an important point is they are mechanically connected to this ryanodine receptor. And the ryanodine receptor is the key calcium releasing receptor. By the conformational change in the dihydroxypyridine receptor induces a conformational change in the ryanodine receptor, and this is the key step for the calcium release that's going to ultimately cause muscle contraction. And this calcium influx through the dihydroxypyridine receptor, which does happen, but it's not crucial for the contraction of the muscle. That's been shown by various experiments. So the key step is this conformational change in the ryanodine receptor that allows calcium to leave the sarcoplasmic reticulum, which is the key calcium store in the muscle, and travel to induce movement in the muscle. So how do we make muscle contraction calcium dependent? Well, it's all to do with these molecules we've outlined that interact with the actin, troponin T, troponin C, and tropomyosin. And so the key calcium binding site in these molecules is troponin C, as we said. And troponin C, through its intimate relation with these other molecules, introduces a conformational change, which induces conformational changes in the whole complex, and the troponin T displaces the tropomyosin that's blocking the myosin-actin binding site. So a binding site is now revealed on the actin molecule where myosin can bind. And so this enables the formation of this myosin-actin cross-bridge. And so that enables the start of this process called cross-bridge cycling. And a key part of this process is the binding of the ATP molecule. And the binding of this, the hydrolysis of this ATP molecule induces the change in the angle of the myosin head group. And then at the release of the inorganic phosphate molecule, we get something called the power stroke. And this is the contractile force that moves the thin filament relative to the thick filament. And this is the ultimate substrate of muscular movement, muscular contraction. So here's just a animation I found that demonstrates this cross-bridge formation between these blue myosin molecules with the globular heads and the actin molecules, and the role of this spring protein titin as well, which is a key protein to consider. So there's some important functional consequences of these processes in terms of how contraction is realized. First of these I want to consider is summation. So if lots of action potentials arrive all at once, then the calcium doesn't return to its baseline level in the muscle cell before the arrival of the next action potential. So this means more calcium can facilitate more muscle contraction, and so we get a summation of the effect of the contraction and so the tension generated in the muscle. And if there's a very strong series of potentials, this can lead to a tetanus, which is a very sustained contraction. Another implication of these processes is this length tension relationship, which refers to the maximum tension a muscle can generate at a particular length. And we have to consider both the active and passive tension. So the passive is just the intrinsic property of the muscle. If you stretch anything, it's going to have a passive tension, but the active tension is to do with the myosin-actin interaction. But a lot of textbooks don't consider the effect of the passive tension it has on the total tension generated, so that's an important point. But the tension generated by the muscle is greatest at the length of the maximum cross-bridge overlap, and that's an important consequence. I think if the length is shorter than that, then some of the myosin heads they can collide with myosin heads from other myosin molecules, and so the tension generated is submaximal. But if we lengthen the muscle too much, the myosin heads won't all come into contact with an actin molecule, and so the tension will be submaximal again. So at the point at which the actin and myosin can form the maximum number of cross bridges based on the number of myosin heads that can bind to an actin binding site, that's the point at which the tension generated is greatest. So if we think about it as a story, and how this message, this action potential that's been transduced from the neuromuscular junction, how that's converted into tension generation, contraction, and movement ultimately. And then calcium, a key step that I haven't quite gone through in the first bit, is that calcium is reaccumulated in the SR by the circuit protein, and that's a key step to end the contraction of the muscle and lead to relaxation. So again, to think about clinical relevance of muscle structure, we think of Duchenne muscular dystrophy, and this relates to another key protein, which is dystrophin, and dystrophin binds the actin elements of the cytoskeleton to the muscular membrane and the extracellular matrix beyond. And it's a key molecule for maintaining the mechanical stability of the muscle. So if we have a mutation in this protein that leads to it not functioning properly, we get a weakness, we get a loss of this mechanical stability, we get muscle weakness, and then muscle wasting. And this is an X-linked disorder, so it's much more common in boys than in girls, but it can have a very tragic effect on people's lives and often is fatal. So coming up with new ways of replacing the incorrect dystrophin molecule, the mutated dystrophin molecule, is leading to things like gene therapy, trying to make cells express a proper dystrophin molecule to prevent the muscle weakness and wasting. And that would be an interesting point to look up if you're thinking of writing an essay on this topic. So now I'm going to think about cardiac muscle, and the key point you've got to think about, especially just in revision or thinking about writing an essay on this topic, think about the key differences between cardiac muscle and skeletal muscle in order to answer that question. Why do we need more than one type of muscle? And so looking at the growth structure of cardiac muscle, it's instantly different to that of skeletal muscle. We see that the cardiac muscle fibers are branched, they're interdigitated, and they have these structures called intercalated discs, which are essentially where the membrane of cells becomes both mechanically and chemically linked. And we'll come to that in a minute. So thinking about the differences between cardiac muscle and skeletal muscle, well, they're both striated muscle, which means they both have this linear structure, these sarcomeres, and they're both, the cardiac muscle is branched and interdigitated, as we just discussed, compared to the linear structure of skeletal muscle. And as we said, these structures called intercalated discs allow both force to be transmitted between fibers, and gap junctions exist in these intercalated discs. So we get spread of ions, charge, and excitation. And this leads to an important effect where the cells contract as a unit. Excitation can pass through cells, and the cells contract together, which is opposed to the motor unit we saw before with muscles. So they do have T-tubules, but they're fewer in number. I'm just going to plug in my laptop because I just run out of charge. Okay, so as I said, they do have T-tubules, but they're not located in the same place. In cardiac muscle, they're at these Z lines. The contraction is more sustained, and as are the action potentials that generate them, and that reflects that. And I'm not going to go into the process of action potential generation today. I think that's more second-year material. But the contraction of cardiac muscle is more sustained. And the excitation-contraction coupling is dependent on calcium influx. So we spoke about the significance of the mechanical link between the dihydroxypyridine receptors and the ryanodine receptors. Well, in cardiac muscle, it's not a mechanical link. It's a link dependent on the influx of calcium that we see through the dihydroxypyridine receptor. And so this process is calcium-induced calcium release. And there's this plateau phase of the action potential that allows time for reuptake of calcium, so prevents tetany, because tetany would be disastrous in the heart. The heart needs to beat regularly and not consistently, and so we have this process to try and prevent that. So again, I'm not going to dwell on this because this is sort of second-year material, thinking about this process and how it might go wrong. But I think the key point when we're thinking about cardiac muscle is excitation spreads throughout the heart. In these areas is going to contract and produce this pushing motion that's going to push the blood out of the ventricles and into the body. So an important consequence of the structure and function of cardiac muscle is this thing called Starlings Law. And essentially all it is a reflection of the length tension relationship we saw in skeletal muscle. They have this the same actin myosin processes in cardiac muscle, so we also get this effect. But we're sort of at the upstroke of it, at sort of physiologically realistic values. We're at the upstroke of this length tension relationship, and that's deliberate because we have this Starlings law, which says that the amount of blood in the ventricle when the heart is relaxed, the end diastolic volume, is proportional, is dependent on the, sorry, let me get that the right way around. The force of contraction is dependent on the end diastolic volume. So the more blood that fills the heart, the more blood that's returning to the heart from the body, the stronger the force of contraction will be to get rid of that blood and spread it throughout the body, which makes sense. So in a closed system, this means that we should expect preload to equal cardiac output, and this will only work if it's a closed system. So an important clinical implication of that is in diastolic heart failure. So in diastolic heart failure, we don't get as much filling of the heart because the ventricle can't relax sufficiently, and that's often due to the fact that heart muscle is stiffer. And reduced end diastolic volume means reduced force of contraction. So often we don't get sufficient force of contraction, and we get reduced cardiac output. Okay, so we've considered the first two types of muscle, and finally I'm going to consider smooth muscle. And this is very different. Cardiac muscle, skeletal muscle, you can see lots of similarities, and there are some similarities with smooth muscle, but in many ways smooth muscle is the odd one out. It's very different, and it's very different in its structure. So it's a non-striated muscle, so it doesn't have these defined sarcomeres, and it's present along tracts, and not exclusively along tracts. We see it in the iris and in the piloerector muscles. There's two key types of smooth muscle. Unitary smooth muscle, as the name is a unit. The cells are connected together by gap junctions, and they can be found ensheathing the organs, such as the airways, guts, and blood vessels. And typically, unlike cardiac and skeletal muscle, the contractions are often triggered by hormonal action, with a modulation by nervous input, typically from the autonomic nervous system. And we can see spontaneous contractions, which gives it some similarity to cardiac muscle, where we get these contractions generated from the sinoatrial node, not based on input from elsewhere. In smooth muscle, we can have pacemaker centers, and we can see spontaneous contractions. Another type of smooth muscle is multi-unit smooth muscle, and this is similar in function to skeletal muscle, but in a similar way to the motor units in skeletal muscle, the smooth muscle cells act independently, and we see this in the iris and the piloerector muscles of the skin. And their innervation, like from skeletal muscle, comes from nervous input without there being spontaneous activity. So what does the structure of smooth muscle look like? Well, it's much more disorganized than in the other muscles. We see actin and myosin still interacting with each other, but they're not in the same parallel relationships. Then the force is not just transmitted in one direction, but it can be in multiple directions. And we see the binding of these to these dense bodies and dense bands on the cell membrane. And these function in a similar way to Z lines, in that they're the point of anchoring for the myofilaments. smooth muscle contractions typically have what we call a basal tone. So even if there's limited input, they can maintain a level of contraction, which can be modulated by the autonomic nervous system. And there's three key types of input, and different smooth muscle in different locations gets different inputs. Some smooth muscle is principally driven by nervous activity, and this includes the smooth muscle in the vas deferens and the arteriolar smooth muscle. Some smooth muscle principally has spontaneous contraction with modulation by these different nervous and hormonal factors. And this includes in the gut. The gut is constantly spontaneously contracting and moving food along the gut in peristalsis. And an important substrate of this is the slow wave activity. And we see the pacemaker activity in the bladder. Some smooth muscle is principally regulated by extracellular ligands, so things like hormones and different bodily factors, and that's not dependent on the extracellular membrane potential in the way that something that receives principally nervous innervation would be. So here's a nice diagram to demonstrate how the three ways in which smooth muscle can get inputs. There's a voltage dependent effect, which you can see in some smooth muscle. There can be ligand gated calcium channels, and there can be ligand gated IP3 G protein coupled receptors that can release calcium from the intracellular stores. But the common substrate of the other types of muscle is the upregulation of calcium and the increase in calcium in the cell. So just gone through that, yeah. Okay, so we've got this raised calcium in the cell as we did for the other two types of muscle. So now we need to translate that into contraction and tension generation. And the mechanism by which this happens is different in smooth muscle than it is in skeletal muscle and cardiac muscle. So in the absence of calcium, we have these myosin light chains that prevent the formation of actin-myosin cross bridges. So instead of tropomyosin being the key blocker of these cross bridge formations, it's now a myosin light chain. So calcium ions, they enter the cell and bind to calmodulin, and this calcium calmodulin complex can bind to myosin light chain kinase and causes activation. So myosin light chain kinase phosphorylates myosin light chain, as the name suggests, and the phosphorylation of the myosin light chain relieves its inhibition of the ATPase activity of the myelin head, which allows cross-bridge cycling to occur. And again, though contraction in smooth muscle is a much sort of slower process in most cases, we still need to be able to reduce contraction in low calcium conditions, and there's a phosphatase enzyme that's important for this. And in some smooth muscle, contraction can be maintained even without calcium, and this is due to a latch state. I'm not going to go into that, but that's something to look at again if you want to write an essay on this topic. So here's the story, like I presented before, for smooth muscle, how we get from these signals. All of a sudden, the signal is not just an action potential in a nerve. It can be a hormonal signal or a neurotransmitter signal. And we convert this into a calcium influx again, but then the process by which we convert that into cross-bridge cycling is again different. So I hope you've had to think about, why do we need the three types of muscle? Because this is the key question that sort of surrounds this lecture. because essentially, why can't we just have one type of muscle that performs all three of these functions? And I think if you're going to write an essay, or if you're going to just revise this topic and try and understand why we have three types of muscle, you need to think about how the structure of these muscles relates to the function. And I think this is particularly apparent for smooth muscle, where we can see for smooth muscle, we don't just want it to be very fast, transient changes in response to depolarization like we might want with skeletal muscle when we're making fine movements. But if we want to induce digestion and induce contraction of smooth muscle in the gut, we don't want that to be a fast process. We want it to be sustained, and we want it to be able to be modulated by different factors. And this is why we can introduce hormones, and we introduce this more sort of sustained element of contraction, a less sort of transient model of contraction, but something that allows contraction to be sustained at a basal tone and modulated, and generating tension in different ways. Okay, so I just wanted to point out some key sources that I used for this to generate this lecture, because they're three very useful textbooks for all of revision, especially for physiology. And they're the Oxford Handbook of Medical Sciences, which is a med student's Bible, and so I'm sure many of you will have come across it, but it's perfect. It's actually based on the first year course here at Oxford, so it's perfect for revision. There's this textbook, which is also perfect for revision, Costanza Physiology. It doesn't necessarily go into great amounts of detail, so if you wanted to go into great detail on a topic, you might want to look at something like Bolton and Bull Peep, but Costanza Physiology is a really great revision textbook. And also this textbook, which I used a lot to revise when I was doing physiology, Ganong's Review of Medical Physiology. Okay, so that is the end of my lecture. So thank you very much. If you have any feedback, I think you have to fill in the feedback form, and if you have any inquiries, don't be afraid to drop me an email.
That was brilliant. Thank you very much, Greg. Yeah, just to reiterate that the feedback link is in the chat, so please do fill that in, and we'll be uploading this to YouTube as well, if you want to catch up on it. Brilliant. Thank you very much.
Cheers. Bye.
Thank you. Bye.